Hello everyone. Today we'll be solving Cambridge International AS and A level Biology Paper 1 multiple choice May-June 2021 Paper 12. Question number 1. Which row describes the membrane surrounding each cell structure? Chloroplast. Single. Well, chloroplasts have double membranes. Mitochondrion also contains double membranes. And nucleus contain double membranes. So option 1, D will be the answer. The mean width of mitochondria in an electron micrograph is 6 millimeter. The magnification of the electron micrograph is 9600. What is the actual mean width of the mitochondria? We know magnification is equals to image divided by actual, and image being 6 millimeter. So 6 millimeter divided by 9600 equals to 6.25 into 10 to the power of -4 millimeter. So B will be the correct answer. Question number 2, option B. Which sequence shows the correct order of some of the stages in the production and secretion of an enzyme? All right. The production and secretion of an enzyme does not start with the Golgi body. It starts with the mRNA, but then after that, it doesn't go to smooth endoplasmic reticulum. It starts with the ribosome, then rough endoplasmic reticulum, then it packs into a vesicle, then it goes to Golgi body. Option C is a possible correct answer. Smooth endoplasmic reticulum and then to mRNA. No, this is also wrong. So option for question number 3, option C. Different antibiotics functions in different way. Ideally, the antibiotic kills the bacteria, but does not harm the infected human. One type of antibiotic, tetracycline, can affect the way in which human mitochondria function. What explains the effect of tetracycline on human mitochondria? Number 1, antibiotic prevents the synthesis of peptidoglycan in cell walls. Option 1 is wrong. Human mitochondria does not have cell walls. Option 2, the antibiotic prevents synthesis of linear DNA. Human mitochondria does not have linear DNA. The antibiotic prevents translation by binding to 70S ribosome. Human mitochondria has 70S ribosome. So D will be the correct answer. Question number 4, answer will be D. A prokaryote cell is 5 micrometer in length. A virus particle is 300 nanometer in length. How many times larger is the prokaryote cell compared to the virus particle? So the virus particle is 5 micrometer, which we can multiply with 1000 to make it nanometer, divided by 300. So that will be 17 times. Question number 5, answer is B. A student carries out a semi-quantitative test with Benedict's solution. Which statement about the procedure is correct? It detects only the presence or absence of glucose. It's a wrong statement. It detects the presence and absence of reducing sugar. It provides an indication of relative reducing sugar concentration. It's a correct statement. It provides a relative reducing sugar concentration. So B is correct. 6B. Which row is correct for carbohydrate? Question number 7. Macromolecule. Macromolecule is, all right, so we know glycogen is macromolecule. Starch is macromolecule. So sucrose and glucose, they're cancelled. Monomers. Sucrose cannot be a monomer. Alpha glucose can be a monomer. Polymer. Glycogen can be a polymer. So option 7, option D. 8. Which row shows the correct general formula for glycogen? Glycogen is formed from glucose, which has reacted by condensation reaction. So glucose has a general formula of C6H12O6. But while forming glycogen, it will give off one water molecule each. So it will become C6H10O5. C6H10O5. Option C is the correct answer. Chitin is a structural polysaccharide found in hard outer shell of animals such as crabs. Chitin is made of amino sugars that contain NH groups. The diagram shows two sugars in the chain of chitin molecule. Which polysaccharide is most like chitin? All right. According to the diagram, if we look at this particular diagram, from this particular point to this particular point is actually 180-degree inverted. So this looks like definitely cellulose molecule. All right. So cellulose. Question number 9C. 4 fatty acids and their formula are listed. You can see caprolic acid, lauric acid, oleic acid, palmitoleic acid. Which fatty acids are saturated? The saturated fatty acids does not have carbon-carbon double bond. So oleic acid and palmitoleic acid, they have carbon-carbon double bond. So must be avoided. So caprolic and lauric acid. A is the correct answer. Question number 10A. Which molecules contain the smallest number of hydrogen atoms? Smallest number of hydrogen atom. We know in alpha glucose, there is a total of, alpha glucose is C6H12O6. So there is 12 hydrogen. A glycine molecule, an amino acid in which the R group is hydrogen. An amino acid where hydrogen is the R group, we can see in that one, there is total 4. There is total of 5. There is 5 hydrogen. 5 hydrogen. Glycerol molecule. A glycerol molecule consists of a CH2OH, CHOH, and CH2OH. So there is a total of 2 plus 3, 4, 5, 6, 7, 8. 8 hydrogens. A saturated fatty acid containing 8 carbon atoms. So saturated fatty acid, all right, first of all, let's draw the carboxylic acid. Containing 8 carbon atoms means there is another 6 CH2 and there is a CH3. Total, there is 8 carbons. So 1 plus 12, 13, 13 plus 3, 16. 16 hydrogen presents. All right, so B will be the correct answer. Question number 11, B. Question number 12. The diagram shows sucrose and sucralose. We can see sucrose and we can see sucralose. Both look similar in shape. However, the OH group here is substituted with the Cl, which is on top. The CH2OH is substituted with the CH2Cl. The CH2OH is substituted with the CH2Cl. We can see these few differences. All right, the rest of the molecule looks the same. The enzyme sucrase breaks down sucrose but cannot break down sucralose. 4 students were asked to suggest why sucrase can break down sucrose but not sucralose. 3 of the students gave correct suggestion. Which suggestion cannot be correct? All right, the question, all right, let's see. The first statement, the chlorine atoms change the shape of the sucralose molecule, so it is not the same shape as the active site of sucrase. We can see the chlorine atom does not necessarily change the shape of the sucralose molecule. All right, so it could be a wrong statement. The next points, let's see and judge them. The chlorine atom of the modified fructose cannot bind to the active site of the sucrase. Yes, this is correct because they cannot, form those bonds, those that are needed. All right, the OH group will be able to form hydrogen bonds, but chlorine will not be able to form hydrogen bonds. The chlorine atom cannot cause an induced feed, so sucralose does not enter the active site of sucrase. Also correct. The chlorine atom can atoms cause fewer temporary hydrogen bonds between sucralose and the active site of sucrase. So the chlorine atoms cause fewer temporary hydrogen bonds. Correct, because once the chlorine is introduced, those portions no longer can produce hydrogen bonds. So this is also correct. So since the question says which suggestion cannot be correct, that is A. The suggestion was the chlorine atoms changes the shape of sucralose molecule. We don't necessarily see the sucralose molecule to be changed in shape. So it is not the same shape as the active site of sucrase. We don't know about it and precise data is not available. So option A is definitely wrong. The graph shows the energy levels involved in an enzyme catalyzed reaction. Substrate molecules X and Y combine to give product Z. Which row shows the reduction in activation energy due to the enzyme? So obviously B shows the reduction in activation energy. So B is the correct answer. Question number 13, B. Which roles of the cell surface membrane results from the properties of phospholipids? All right. Results from the properties of phospholipids. To allow cytokinesis to occur in mitotic cell division, to allow entry and exit of oxygen and carbon dioxide, to allow the phagocytosis of a bacterium into a cell. So we know bacteria can be phagocytosized by using cell membrane. That is correct. Oxygen and carbon dioxide can diffuse in and out because it is nonpolar. And cell membrane also allows mitotic cell division when cytokinesis occurs. So both 1, 2, 3 options are correct. Question number 14A. Question number 15. Which factors can be changed to affect the rate of facilitated diffusion across a cell surface membrane? Number 1, the surface area of the membrane. All right. Okay. Surface area of the membrane, if it is increased, rate of facilitated diffusion will also increase. If concentration gradient is increased, rate of facilitated diffusion will also increase. If the number of specific protein channels, those that are needed for the facilitated diffusion, are increased, rate of diffusion will increase. So 15A. Question number 16. The diagram shows an experiment using a model cell to investigate the movement of substances. All right. Membrane permeable to monosaccharides and water. Model cell. Solution containing 0.03 mole per dm cube sucrose, 0.02 mole per dm cube glucose. Solution containing 0.01 mole per dm cube sucrose and 0.01 mole per dm cube glucose. Which statements are correct? There is a net movement of sucrose out of the model cell. Sucrose cannot pass through the partially permeable membrane. So option 1 is wrong. If option 1 is wrong, then A, B, C all cancelled. Option D is correct. 16D. Human chromosomes have different parts. Some parts are more numerous than others. Which parts are listed in order from most numerous to least numerous in a human white blood cell? All right, centromere, then nucleotide. Obviously, nucleotide is more higher in number, so wrong. DNA molecule, then telomere. Obviously, telomere will be higher in number compared to DNA molecule. So wrong. Histone protein and telomere. Okay, so there will be more number of histone protein than telomere and less number of DNA molecule, obviously. So C technically should be the correct answer. 17, the answer is C. The diagram represents the cell cycle of human cell. During the cell cycle, the number of chromatids changes. Which row is correct for the number of chromatids in M, G1, and G2? M, there is a total of 92 chromatids. We know during mitosis, 92 chromatids are present. In G1 phase, there will be a total of 46 chromosomes present. However, in G2 phase, then it will again double up because in the S phase, there will be DNA replication. DNA replication will occur here. So there will be 92. Option C. Question number 18, option C. A single skin cell was isolated and transferred to growth medium in sterile petri dish. The petri dish was incubated for 16 days. During this time, the number of skin cells multiplied as a result of repeated mitotic divisions. One of the chromosomes in the nucleus of each skin cell has telomere that contains many repeats of base sequence TTAGGG. On each of 4 days, 4, 8, 12, 16 of the incubation period, a single cell was removed from the petri dish. The total number of bases in the telomere of this chromosome was determined for each cell. Each of the 4 cells had a different total number of telomere bases for the chromosome: 5548, 5580, 5645, and 565700. What was the total number of telomere bases in the chromosome from the cell that had undergone the most mitotic division? Any cell that undergoes the most mitotic division, telomere bases are going to decrease. The smallest number in all of this is 5548. So A should be the correct answer. 19, option A. A short question number 20. A short piece of DNA, 18 base pair long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strand. Some of the results are shown in the table. The number of nucleotide bases, strand 1, strand 2, information like cytosine and thymine, one of the information is provided. How many nucleotides containing thymine were present in strand 2? All right, we know that the DNA is 18 base pair long. So there is a total of 18 DNA on this side and 18 DNA on the other side. So if there is in the strand 1, there is 4 cytosine, then in the strand 2, there will be 4 guanine. And if in the strand 2, there is 5 cytosine, in strand 1, there will be 5 guanine. Similarly, if there is 7 thymine in strand 1, there will be 7 adenine in strand 2. All right, so the total number, 7 plus 5, 12, 12 plus 4, 16, 16, 18 minus 16 is equals to 2. So this will be 2 and 2. All right. How many nucleotides containing thymine were present in strand 2? Thymine 2. A is the correct answer. Question number 20, A. Question number 21. The diagram shows a strand of DNA and mRNA during transcription. All right. Which row correctly identifies 1, 2, 3? All right. So between, first of all, number 1 is cytosine. Cytosine is a pyrimidine. All right. And we can see this is between DNA and mRNA. So, all right, number 2. All right, number 2, which is guanine. All right, which is guanine, definitely should be ribose because this is the mRNA. All right, and then, all right, number 3 is between the guanine and cytosine, there will be 3 hydrogen bond. All right, so D is the correct answer. Question number 22. Two students were discussing the involvement of DNA and RNA in transcription and translation. Student 1 always stated correct facts. Student 2 gave further information which was sometimes correct. A length of mRNA is 747 nucleotide long, including stop and start codons. The start codon codes for amino acid, but then the stop codon does not code for amino acid. So it will be 747 minus 3 divided by 3. So the answer is 248. So the mRNA can produce a polypeptide that is 249 amino acid long. This is a wrong statement. If point number 1 is wrong, then A is incorrect. All right. For number 2, adjacent mRNA codons of AAU and CUG bind to complementary tRNA anticodons. Okay, there is a total of 14 hydrogen bonds formed between the two codons and their anticodons. We know A produces 2 hydrogen bonds and U also produces 2 hydrogen bonds. So total there will be 6 here. And C produces 3 hydrogen bonds, U2, G3. So there will be a total of 8. So 6 plus 8, total will be 14. So this is a correct statement. Option 2 is correct. RNA polymerase catalyzes the formation of mRNA from the template strand of DNA. All right. During translation, an RNA adenine nucleotide will pair with a DNA thymine nucleotide during translation. All right. So translation does not involve a RNA adenine, binding up with the DNA thymine. This is a wrong statement. All right. mRNA will bind up with tRNA. So number 3 is wrong. All right. So we have got only 2 correct. Now let's see whether number 4. So technically number 4 shouldn't be correct. Now let's see. A DNA adenine nucleotide is structurally different to RNA adenine nucleotide. All right, it's different. The difference is the hexose sugar. DNA is, well, the difference is not a hexose sugar, it's a pentose sugar. All right, so number 4 is also wrong. So 2 correct only. Question number 22 is C. Which descriptions of the adaptation of xylem vessel elements to their functions are correct? They have pits between vessels, allowing water to pass from one vessel to another. So xylem, yes, they have pits between them, one vessel to another vessel, they can allow water to pass. They are thickened with lignin to stop them collapsing from the column of when the column of water is under tension. Yes, they are lignified, which prevents them from collapsing. They form a continuous hollow tube through the plant, providing little resistance to water movement. Yes, they do all of these 3 things. So 23 option will be A. The diameter of a tree trunk usually decreases slightly during the day. Which changes in environment factors during the day could cause the diameter to decrease even more? All right, increased humidity means less transpiration. Increased wind speed means more transpiration. Increased light intensity means more transpiration. So B should be the correct answer. Question number 25. A student wanted to find out the rate of transpiration using a potometer as shown. There is a cut chute, there is a reservoir of water, air bubble, capillary tube, and then it is placed in a beaker of water. The student was told to work out a value for the transpiration using the unit millimeter cube meter square and hour per hour. Which measurement would the student need to take? The volume of water in the reservoir. It's not important what volume of water in the reservoir is present. So this is totally wrong statement. All right, let's see, move on. Distance the air bubble moves. Yes, we need that. The radius of the capillary tubing. Yes, we also need that. We need the time because we need per hour. Okay. And the surface area of the leaves. We need all of this information. So for question number 25, option C. Question number 26. In plants, assimilates such as sucrose are loaded from source cells into sieve tube elements. In plants, assimilates such as sucrose are loaded from source cells into sieve tube elements. The assimilates are then transported from source to the sink. What is needed to transport sucrose from source cells through phloem tissue to sink cells? All right. What is needed? So there is a water potential gradient between the source and the sink. All right. Okay. All right. Source to the sink. All right. So water potential gradient between the source and the sink is not needed. All right. However, a hydrostatic pressure gradient between the source and the sink is needed. And ATP is needed. So option C will be the correct answer. 26, option C. Question number 27. The sinoatrial node initiates cardiac cycle. What is the correct order in which the chambers of the heart contract? All right. Both atria and both ventricles, they contract together. So 27, option B will be the correct answer. Question number 28. The graph shows pressure changes in different parts of the heart during a mammalian cardiac cycle. W, X, Y, Z indicate when a valve closes or opens. In W, a valve closes. In X, a valve opens. In Y, a valve closes. And Z, a valve opens. So W will be closes, X will be opens, Y will be closes, Z will be opens. C is the correct answer. Question number 28, C. What can combine with the heme group of a hemoglobin molecule? We know the heme group can only combine with oxygen, and it can combine with carbon monoxide. Carbon dioxide cannot directly combine with the heme group of the hemoglobin molecule. All right, so 1 and 3 will be correct. Option B is correct. What is the main reaction occurring in blood capillaries at gas exchange surface in a human lung? Carbonic acid dissociates into carbon dioxide and hydrogen ions. Yes, it dissociates into carbon dioxide and hydrogen ion. Option number 30, A. Question number 30. What is the main reaction occurring in blood capillaries at the gas exchange surface in a human lung? A. Carbonic acid dissociates into carbon dioxide and hydrogen ions. All right. Okay, that is not the main reaction that is occurring in blood capillaries at the gas exchange surface. Carbonic anhydrous converts carbon dioxide into hydrogen carbonate ion. No, it is done in red blood cell when it is near the actively respiring tissue. Carbon dioxide combines with hemoglobin to form carbaminohemoglobin. No. Finally, carbon dioxide is produced from hydrogen carbonate ion by carbonic anhydrase. Yes, D is correct. Carbon dioxide is given off in the lungs. 30D. Question number 31. Which reactions take place in blood that is passing through active tissue? Passing through an active tissue, definitely hemoglobin will lose oxygen. So 1 is correct. All right. When H plus ion from the carbonic acid will attach itself to the hemoglobin, oxyhemoglobin, hemoglobinic acid and oxygen will be released. That is also correct. 1 and 2 correct. All right. If 1 and 2 correct, then C, D options are gone. All right. Hydrogen carbonate plus H plus ion, all right, produces H2CO3. This reaction is a reverse reaction. The reverse reaction occurs in the active tissues. So number 3 is wrong. So if number 3 is wrong, B will be the correct answer. 32. Which row shows the tissue that are present in the cell wall, in the wall of the trachea and in the wall of the bronchus? All right, wall of the trachea and bronchus both contains cartilage. So cartilage will be correct. None of them contains squamous epithelium. Squamous epithelium is only found in the gas exchange surface, basically in the alveoli. Yes, they contain goblet cells. So 32, option C is correct. In some cases where a person has lung disease, the partial pressure of oxygen in the pulmonary vein is less than the partial pressure of oxygen in the alveoli. In some cases where a person has lung disease, the partial pressure of oxygen in the pulmonary vein is less than partial pressure of oxygen in the alveoli. What could explain the difference in partial pressure of oxygen? All right. A high proportion of the alveoli are collapsed and do not have enough alveolar capillaries. All right. So yes, this happens. This happens in emphysema. So option number 1 is correct. The partial pressure of oxygen in the pulmonary artery is lower than in the alveolar air. Well, this is a wrong statement. In the pulmonary artery, the percent pressure of uh, oxygen being lower than in the alveolar air uh, does not explain why the pulmonary vein is less. All right. Yeah, is less. So number two, it cannot explain that statement. All right. The rate of diffusion of oxygen from the alveolar air to the surrounding alveolar capillaries is too slow. Yes, number three is definitely correct. So 1 and 3 correct. That means option C. 33, option C. 34. How many times must a molecule of carbon dioxide pass through a cell surface membrane as it diffuses from plasma through a cell in the capillary wall in the alveolus? So let's look at the capillary. All right. So from the red blood cell, 1, 2, 3, 4, 5. How many times a molecule of carbon dioxide must pass through a cell surface membrane as it diffuses from the plasma? So from the plasma, if this occurs, then there will be 4. Total of 4. D is the correct answer. Question number 34, 4. D. 35. Which disease is spread by a vector? We know that AIDS is not spread by a vector, cholera not, and then measles not. So C, malaria. It is spread by female Anopheles mosquito. Which statement explains why one dose of vaccination program for measles has not yet eliminated the disease? All right, many infants under the age of 8 months have passive immunity. No, this cannot be the reason. Some children need several booster doses to develop full immunity. This is the main reason why we still couldn't eradicate measles. So option for 36, option B is the correct answer. What, question number 37, what has contributed to the increase in antibiotic resistance in bacteria? An increased use of vaccines for animal diseases? Well, no. Number one is wrong. If number one is wrong, then for question number 37, option D is the correct answer. What explains why monoclonal antibodies can be used to target cancer cells? All right, cancer cells have different antigens from normal body cells. Yes, they have different antigens from normal body cells. Specific cancer drugs can be attached to the monoclonal antibodies. All right, that this does not explain why we can target cancer cells. They secrete a type of antigen that binds to a specific antibody. No, they are secreted by hybridomas of cancer cells and B lymphocytes. No. So option number 38A. Cancer cells have different antigens than normal body cells. Which, question number 39, which statements correctly describe lymphocytes? Each B lymphocyte has the ability to make several types of antibody molecules. No, number one is wrong. If number one is wrong, then option D will be the correct answer. A country introduced a measles vaccination during measles epidemic. Later, it was realized that the vaccinated children were more likely to survive childhood than the unvaccinated children, even when there was no measles epidemics. The vaccine had given the children some protection against the pathogenic infection. What statement could account for this extra protection? B lymphocyte produces memory cells which gave the children passive immunity to this infection. No. Memory cells produced plasma cells which secreted anti-measles antibodies that bound to antigens that are close to resemble measles antigens. Yes, this could be a possible thing. Memory cells produce plasma cells which secreted anti-measles antibodies that bound to any antigen. No, it can't bind to any antigen. All right, then there would be like no diseases at all. T lymphocyte produced memory cells which gave the children natural immunity against these other infections. No. All right, so only B can be correct. So question number 40, option B. Hello, guys. We are regularly posting videos of question paper solving. So, if you want any question paper that you want us to solve related to chemistry and biology, which the channel name suggests, please let us know in the comments. And definitely, we will try to get your question in the queue, question paper in the queue, and we will definitely solve it. All right. Thank you. Subscribe to the channel. Bye bye.